Hey, this is our video on glucogenic amino acids. So taking amino acids, removing the amino group off of the amino acid and either just leaving a nitrogenous waste or forming, using it to transaminate another protein to make different sorts of proteins. But leaving in the wake of that, so this deaminated amino acid, eventually undergoing other reactions that will give you pyruvate, which can then be converted into glucose. So if you want to think about when you would be doing this, this would be if you were in a starvation mode. So it doesn't have to be necessarily a very severe starvation mode, but it would be a non-fed state. So you are not having a lot of blood sugar, you haven't eaten for a while, even when you're sleeping, this is happening. So your energy levels are low, you're not running the Krebs cycle through glycolysis first because you have lower blood sugar. So you need to raise your blood sugar. You'll be using a lot of fat, preferentially fat for energy creation, but you will also be doing some breakdown of amino acids for energy creation. Now we've seen amino acids turning into ketones, which we'll talk about more in this unit as well, but here these amino acids, these specific glucogenic amino acids, are going into pyruvate and glucose. So what we're looking for is just how do we get it into pyruvate first? How do we take that amino acid and turn it into pyruvate? Then gluconeogenesis is very easy to do from pyruvate, so we know how to do that. We've talked already about alanine and how we can get glucose out of that from the skeletal muscles and into the liver and things like that. But each specific glucogenic amino acid is going to have a different way that it gets turned into pyruvate. But once it's pyruvate, then it's all the same. Then you're just going to do just straight up standard gluconeogenesis. Now, if you look over here, at this chart here, we have, proteins are made up of a lot of amino acids. We're using glutamate here as an example, and we've seen alpha-ketoglutarate a gabillion times already, and we just know that with transamination, we're just bouncing an amino acid back and forth between glutamate and alpha-ketoglutarate, and that will give you, various products that you can use in various circumstances. Like we've seen this in the malate aspartate cycle. We know alpha-ketoglutarate is part of the Krebs cycle, etc. All of that. We've also seen how we can link this with alanine here, so we can get alanine from pyruvate that came from glycolysis. So here we're, going kind of in the backward cycle. It's the glucose-alanine cycle again. And then once we have alanine, then we are taking it out of the muscles, say, and transporting it through the blood into the liver, where we can do gluconeogenesis. So we convert it back into pyruvate and back through gluconeogenesis. So we've talked about all of that already. What we haven't talked about is another direction we can go, where it's just really the reverse here, where we have glutamate into alpha-ketoglutarate. Here we're taking alpha-ketoglutarate and converting it to glutamate. So again, just transamination here. And then we're dehydrogenating it. So we're removing hydrogens, water essentially. We're taking that off, and that's giving us this NH4. And that's going to lead us to something called the urea cycle, which we're going to talk about in coming times. The main gist of this is to look at the nitrogen part. So when we are deaminating and transaminating, we are moving, an amino group that has nitrogen. So we can end up with nitrogenous waste if we are deaminating. If we're transaminating, then that nitrogen is part of the amino group. It's just bouncing from one protein to another. But when we're deaminating, like we're doing for gluconeogenesis, we can end up with these nitrogenous wastes. And this will lead us to having to deal with that nitrogenous waste. For example, the urea cycle. If it were NH3, that would be ammonia, and that would be dealt with by the kidneys excreted in the urine, as is urea. Urea is also excreted in the urine as well, but also the urea cycle is a way of converting it into other things. So we're just really watching where that amino group is going, if it's being thrown away as a waste or if it's being, put onto something else in transamination. Okay, so different amino acids, so glucogenic amino acids and how they get to pyruvate. One, alanine, just simple transamination, bouncing the amino acid from one place to another, and we end up with pyruvate. And then you can do, anytime as soon as you have pyruvate, you can do gluconeogenesis and create glucose and raise your blood sugar. Glycine is another glucogenic amino acid. Here it's an enzyme called a transferase that is going to be working on the glycine. It will create an intermediary called serine, and then we use serine dehydratase to form pyruvate. Now when you use a dehydratase, there goes water as a byproduct. In this case, it's being created as a byproduct, not transferred to another molecule, and then boom, there's our nitrogenous waste. This is ammonia. This will be dealt with in the kidneys as it goes into the urine. And don't worry about these little byproducts here. We don't have to worry about those. We're just watching really, the main point here is that we are changing the molecule, taking the amino acid away, creating pyruvate, and we're going to end up with gluconeogenesis happening. Here, cysteine. Cysteine is going to be reduced, so adding an electron. So here is where you're using NADH. We've really been talking a lot about making NADH and using it in the electron transport chain. But here we're using the hydrogen from the NADH and we're regenerating NAD+, which is good. That can be used when we start glycolysis up again, assuming your starvation cycle doesn't last for too long, right? And normally it doesn't. So you need NAD+ ready for when you start running, the electron transport chain, when you start doing glycolysis and all that kind of stuff again. You need to have a nice steady supply of that. But what it turns into then is cysteine. Cysteine is something that is a few uses. If you remember, it was part of some various fatty acid processes as well, or antioxidant processes like the NRF2 with creating endogenous antioxidants. So cysteine has some uses. All of these things have uses within the cell other than just breaking them down into pyruvate. So it's not the best thing to be breaking down your amino acids into pyruvate. It's better to use fats, but you will be doing some of this anyway. Then you have a series of reactions. There's first transamination and then desulfurization, so taking a sulfur off of the cysteine, and then you end up with pyruvate and you can do gluconeogenesis. And then threonine, with threonine there's a couple of different ways this can go. If you're using a dehydrogenase enzyme, you end up with pyruvate. If you use a lyase enzyme just breaks chemical bonds using a process that doesn't have anything to do with oxidation. It doesn't have anything to do with hydrolysis, which are kind of typical ways of breaking bonds. But a lyase just breaks bonds in different ways. So it's breaking the bonds of the amino group, and here you end up with ammonia. It's a toxic byproduct there. And then you get alpha ketobutyrate. From there, you do, again, a bunch of reactions, including one involving a dehydrogenase enzyme and then a carboxylase enzyme, and that gives you succinyl-CoA. And where do you see succinyl-CoA, or where we use succinyl-CoA, is in the Krebs cycle. Always put that apostrophe there. There's no apostrophe there. So we're using succinyl-CoA in the Krebs cycle, which will be handy when we go back into the fed state and we start running glycolysis in the Krebs cycle, or when we're getting acetyl-CoA, out of lipids and running the Krebs cycle that way. You still need to have your succinyl-CoA. now this is the process of turning cysteine to pyruvate. So remember we saw this, this is like the short version. Here's the more spelled out version, and why this is more spelled out here is that we have alpha-ketoglutarate here. So I just wanted you to notice that alpha-ketoglutarate, remember that's from the Krebs cycle, it's from the malate aspartate cycle, all of that, and glutamate, we've seen that a gazillion times. These are all involved in the transamination process of taking cysteine into this mouthful of a molecule. And then we're going to do the desulfurization, which happens here. You'll notice that we just took a water and we added it, or we used it to essentially break the sulfur right there off of that. And now we have pyruvate, and we can do gluconeogenesis. Okay, so the whole point of all of the glucogenic amino acids is that we are getting pyruvate, and we are going to be able to do gluconeogenesis from that. And that will raise our blood sugar. And that will allow cells to do glycolysis again. And therefore the Krebs cycle. So just to kind of think of this from a big picture point of view, just picture yourself sleeping. You haven't eaten for a while. You have run out of your ready stores of glucose. You're starting to do lipolysis. You're breaking down your fats, and you're doing beta oxidation, and you're running the Krebs cycle from the acetyl-CoA that you get from there. You're also starting to break down your proteins, especially in skeletal muscle, and you're doing the same thing. You're getting ketones out of both the beta oxidation and from breaking down certain proteins. And then from these other types of proteins, a glucogenic amino acid, you're getting pyruvate, so you can do gluconeogenesis. So you're still running the Krebs cycle from acetyl-CoA that comes from breaking down fats, so beta oxidation, and also from ketones, from ketogenic amino acids, so when you're in the starvation state, you're not, your cells aren't going to run out of energy unless it lasts for a very long time. They're able to break down the fats and the ketogenic amino acids. You get the ketones, you get acetyl-CoA, and you're able to run the Krebs cycle. But your blood sugar is low, so you don't have that quick, easy source of energy. So the glucogenic amino acids are going to help to raise your blood sugar so that you can do glycolysis again, and by giving you pyruvate so you can do gluconeogenesis. Then at some point you're going to eat, and your blood sugar will go up even more, and you'll switch away from breaking down fats, breaking down proteins, both kinds of proteins, and you'll switch more to making those things instead and using the glucose for energy. So it's just a balance, all in response to your blood sugar levels and what your energetic needs are. Now for pyruvate to glucose, this is gluconeogenesis. So we've seen this before, so we don't need to spend a lot of time on this. It's really essentially glycolysis backwards, because glycolysis is in most stages reversible. So that's really all you're doing. You're taking pyruvate, you're using ATP, so that uses energy, and then you end up with ADP, with a phosphate broken off. So we know that ADP is ready when we start running, glycolysis again in the forwards kind of way. We know that ADP is there to accept a phosphate in order to create ATP. But here we're using the energy, so we're breaking apart the ATP. That gives us oxaloacetate. It's also part of like the last step of the Krebs cycle. We're going to use some more energy here in the form of GTP. So we're breaking GTP. So that is where we use energy. And we get GDP, which can accept a phosphate in other processes, right? So back in the Krebs cycle, we used some GDP and added a phosphate to it to get GTP. And then here we are at the end of glycolysis, right? If we were running in the forward direction, this would be the end part of glycolysis, and we just run it straight up backwards until we get to glucose, just glucose. And we can, use that glucose to raise our blood sugar. Okay, so just to reiterate the whole why you're doing this, why you're breaking down those amino acids, is just so that you can continue to have blood sugar and energy while you are not eating. So your blood sugar drops, you're breaking down your fat, you're breaking down your proteins, using these things for energy. And remember, breaking down the proteins isn't great because you're losing a little bit of muscle mass. So every night you lose a little bit of muscle mass, but just a little bit, right? And you can build it back up during the day. You wouldn't end up getting a net loss of muscle mass, like a noticeable, real, consistent amount of muscle mass loss, unless you were consistently in a starvation or fasting phase. Now, the trigger mechanism, so the way we regulate this is that as you're doing a lot of beta oxidation and, making a lot of, ketones and pyruvate out of breaking down your amino acids, you're getting a lot of acetyl-CoA. And acetyl-CoA is something that inhibits glycolysis. So when you inhibit glycolysis, that's something that we do when we have, a lot of energy, right? But we're also doing it when we don't have a lot of energy, if that makes any sense. The whole thing is that when you have not enough energy, you don't want to do glycolysis because you don't have glucose handy, right? You want to run it backwards so that you can create more glucose. When you have a lot of energy, on the other hand, so you're making a lot of acetyl-CoA, whether it's from glycolysis or wherever it's coming from, then you don't need any more energy. So you want to start pushing things more into the storage thing. Really just think of when you have acetyl-CoA from whatever source, whether it's coming from glucose or whether it's coming from fats or from lipids, that means that you're going to be inhibiting the breakdown of glucose from wherever the acetyl-CoA came from. So that will be where it's, depending on whether you're in starvation mode or in the fed mode. Now, other things that inhibit creating acetyl-CoA out of pyruvate are pyruvate dehydrogenase being inhibited by glucagon. So we know that glucagon is something that you release. It's a hormone that you release when your blood sugar is low. So it helps to encourage gluconeogenesis by inhibiting pyruvate dehydrogenase. Cortisol, same thing, hormone released during stress, of which starving is stressful. So, you release cortisol during stress. Now, not when you're sleeping, right? You're hopefully not releasing cortisol while you're sleeping, but to wake up, you do release cortisol. Cortisol is released just before you wake up, about an hour or so before, and it helps you wake up, which is good. But it's also pushing you towards gluconeogenesis, which is also good because you're about to get up and you're going to need faster energy supplies as you start to move around. Also, adrenocorticotropic hormone. Adrenocorticotropic hormone and cortisol are linked because adrenocorticotropic hormone stimulates cortisol. So that those two are working together as synergists. But the whole point is just to push pyruvate back into gluconeogenesis so that you are making glucose rather than burning up whatever small amounts of glucose you have for the Krebs cycle. Okay, now the early fed state. This means you just ate and you're absorbing nutrients. So you're just starting to absorb nutrients. And we know that it takes food a bit of time in your stomach to break down and get into your intestines. And that can take some hours. Some of the more simple sugars can be absorbed much more quickly than a few hours after eating. But it is not like you eat your breakfast and all of a sudden, boom, your blood sugar is up as high as it'll go, and you're just have all those nutrients already on hand in a minute. Also, your body takes a bit to respond to stimuli. It's not just an instant switch. So it's going to take you a little while to go back to running glycolysis and pushing the Krebs cycle forward after you've been in that fasting or starvation state and you've been sitting there or laying there sleeping and breaking down your fats and your proteins. Now you're not having glucagon out. So glucagon isn't there to inhibit pyruvate dehydrogenase and push glycolysis back towards gluconeogenesis. But now you have insulin coming out. Insulin is something that you release when your blood sugar is rising. It pushes your glycolysis pathway more towards glycogenesis rather than straight up glycolysis. So instead of just using energy, this is where you're going to be, or creating energy, you're just going to be storing that glucose for later. Now, in your muscle cells, the pyruvate that you've been making but not running through the Krebs cycle, that turns into lactate. And lactate can go to the liver and it can get turned back into pyruvate in the Cori cycle. And from there, you take the pyruvate and you do gluconeogenesis. So here's the reaction for turning pyruvate into lactate. Not super complicated. You just have this dehydrogenase enzyme and you use it on pyruvate and you get lactate. And it goes reversibly too. So you can just, in the Cori cycle, you'll take lactate, use the same enzyme, lactate dehydrogenase, just, make that backwards too. The whole thing is backwards and we end up with pyruvate. Also remember that lactate is also lactic acid and can lead to lactic acidosis if you have too much of it. That tends to not happen while you're just at rest and experiencing a fasting state, but it would happen more when you're in a situation of you're exercising and you're using a lot of energy or creating a lot of energy, but you run out of oxygen, so you can't, you can't run your Krebs cycle, so the pyruvate builds up and you end up with too much lactate. So that would be intense exercise for that. Okay, so we know how we got lactate. So we got, we turned our pyruvate into lactate, and this is happening in skeletal muscle, right? So over here, we have the skeletal muscle and other types of cells too. It's not just in muscle cells. And over here, we have the liver, a little bit in the renal cortex, but specifically your hepatocytes are the most, active for running the Cori cycle. So this would be the Cori cycle happening over here. So glycolysis happens, right? So we're making our lactic acid here by creating our pyruvate, and then we're not running the Krebs cycle. So when the Krebs cycle isn't running, we end up with lactate. That gets into the blood, travels through the blood, and it gets to the hepatocytes, and we do the reaction backwards, and we end up with pyruvate, and then we go into gluconeogenesis from the pyruvate, same as how we did it from the pyruvate earlier in this lecture, and then we have glucose, and the glucose goes into the blood, and we end up with raising our blood sugar, so we have an increase in blood sugar, and then that can just be used by the cells again to run glycolysis, make more pyruvate. Either the pyruvate will be used in the Krebs cycle to run the Krebs cycle in the electron transport chain, but if you run out of oxygen or if you go into a starvation mode, then we just keep running the Cori cycle. Okay. Okay, so that is it for our gluconeogenic amino acids, or glucogenic amino acids.